Hello there and welcome back to Unit 6, and as we continue kind of the midsection of the class and more scientific concepts and key things with regard to animals in a non-specific, species-specific approach. This is Reproduction and Insemination, which is chapters 10 and 11 in your textbook. I hope you're reading and following along in your notes in your textbook. I'll go through the objectives. We're going to focus specifically on the female organs of reproduction and their function first. And as number one, the male is a lot simpler. And number two, there's not as much emphasis and effort put into manipulating what happens in the male. So let's talk about the female more specifically. When we talk first about the ovary is responsible for producing both the ova or the egg and the female sex hormones, which is estrogen and progesterone. Very, very critical. We start to get down our hormones and their function and where they're produced. So if there's notes to take as you go along this section, really focus on what we talk about through here with regard to hormone function. We got to go a little bit deeper in the ovary and talk about folliculogenesis, so making the follicle or the ovum, that developing egg that's coming along. So each follicle contains an egg. That development starts deep within the ovary with what we call a primordial follicle. As that wave of follicles develop, each one of them produces more estrogen, which helps mature that follicle as it comes along. Every follicle goes through what we call a stage of recruitment, selection, and dominance. So there's a kind of a whole group of primary follicles that will be developing along, and only a few of those will then become what we call secondary follicles that are the largest, most nourished, healthiest follicles that are coming in that group. There'll be some further recruitment in what we call then a tertiary phase, tertiary follicle. That would be even a fewer selected eggs that might reach maturity in order to help determine which one's going to be ovulated. The Graafian follicle, or the ovulatory follicle, then is a single follicle. We kind of get rid of let's say that we have three tertiary follicles coming on the ovary. There's only going to be one ovulatory follicle, and that will be the Graafian follicle. That will be the largest, most nutritious, that we've put all our groceries into one sack, so to speak, that will then be ovulated when the animal comes into heat. Then that follicle will go through a degenerative phase, or what we scientifically call atresia, where that site where that follicle was ovulated from, or if it never does get ovulated because the animal's pregnant, you'll have a degenerative phase where that follicle will be absorbed back into the ovary. Again, only the Graafian follicle is ovulated, and when ovulation occurs, the follicle ruptures and releases the egg into the infundibulum and down to the fallopian tubes, where we hopefully get fertilization. After ovulation, the follicular cells form what we call the corpus luteum, which is what we're going to talk about next. The CL, or corpus luteum, also known as a yellow body or a callus on the ovary, is what forms there and actually does look like, if you've never felt one or seen one, it looks like a big yellow callus on the edge of the ovary. And what it does is, as it's there and active, it makes the hormone called progesterone. As long as the animal has a functioning CL on the ovary, she's going to think that she's pregnant, and that will maintain pregnancy through term, hopefully. I've got a video link here that you guys can go check out, and I'll let you do that on your own time. But progesterone, very important, developing from the CL on the ovary. The oviduct then is next, and like I said earlier, that kind of collects the ovulated ovum or the ovulated egg into the infundibulum. Think about the ovary is more or less suspended, free in the body, and then the infundibulum is almost like a catcher mitt that sits there, a first baseman's mitt that sits there right around the outside of the ovary and kind of funnels that ovulated egg into the oviduct and guides that egg down there. The oviduct is the site of fertilization for virtually all species. Now, here's where we get some differences. When we talk about the site of fertilization in the pig, that's in the oviduct, and those embryos basically will just scatter throughout the uterine horns, and and basically implant right there. If we talk like the cow, where we only have generally one ovulated egg, after about 3 to 5 days, we're going to have implantation as that embryo migrates to the uterus, hopefully successfully implanting there and growing in the uterus from there. Again, as we keep going down the down the reproductive tract and get to the uterus, that's the site generally of uterine of embryo attachment and development then to full term of each of those animals. So some attach in the horn like the sow, and others attach in the body like the cow or the mare. The uterus is guarded by a structure that we call the cervix, or I've put the gatekeeper. That's a good way to kind of think of it if you're looking for another term, can't remember cervix well. The cervix is guarded or guards that uterine environment, kind of closes down tightly to make sure no bacteria or anything that might damage that developing fetus from passing into the upper reproductive. The vagina is then the passageway, the female organ of copulation, and then environment changes depending on the stage of the reproductive cycle. If an animal is not in heat, it's not a full dry environment, but there's not much going on there either. If an animal is actively in estrus, you'll find it engorged, full of mucus, kind of helping foster the passageway of sperm up into the upper reproductive tract. The urethra from the digestive system also dumps in right at the end of the vagina, so you'll see kind of a dual purpose. Here is a side view cutaway of the reproductive organs of the cow, and notice we start at the vulva at the back and into the vagina, and then right about the pelvic shelf you'll see the cervix sitting there, and then on anterior from that, or towards the head of the cow, you'll see the uterus, and then the uterine horns or the oviducts tend to curl underneath the uterus, and the ovaries sit right underneath there. So as you're palpating an animal or inseminating an animal, you're going to pass your arm up the rectal cavity, and then you'll be able to feel through the soft tissue and palpate the reproductive tract if you're artificially inseminating or a veterinarian is there checking for the presence of a fetus. Here's a top view of both the cow on the left and the sow on the right. Notice the differences in the sizes of the vagina, the length of the cervix, and the type of uterine horns. Notice in the sow there's a long uterine horn. It's fairly narrow. In the cow they're fairly short uterine horns, but the uterus is much larger, and where the ovaries actually sit on each of those. Here's a microscopic look at an egg that's been ovulated there in the center, and then you can see little sperm attached on the outside. The sperm are minuscule in comparison to the size of the egg. So just a nice microscopic diagram here of what's going on when you look at the size of an egg versus a sperm cell. Okay, this is a developing follicle here that's on a cow's ovary, and so you can kind of see the black ring of cells on the outside, and then you see a big area in which there's kind of a void there in the center where it looks like there might be fluid, and then you kind of see another ring of another circle within the circle here, so to speak, towards the bottom middle of your screen, and then another voided area. That's where the actual egg is. The follicle with all the fluid is the large ring, and then fluid maintaining it, and then you actually have the ovulatory egg right in the middle. Another diagrammatic component here that your textbook provides, and I like this one because it shows the maturation process of developing follicles. So if you start at the bottom left-hand side of the ovary, you see the primordial follicles, that then we see some oocytes coming along, a tertiary follicle. Once you get to the top of the screen, then a dominant or Graafian follicle then develops, where we just have one singular large one that's coming along. That follicle, as you get to the right side of the screen, then ovulates, and you see the egg being released, and then that thing forms into a corpus luteum, that yellow body, that callus structure that then protrudes from the ovary there and produces progesterone to tell that female that she's pregnant for a little bit of time. that thing will eventually regress, either if an animal is not in fact pregnant or calves or goes through parturition, becomes a corpus albicans and is resorbed. I said we wouldn't spend very much time on the male reproductive organs and male reproductive tract. There's just a couple of slides here, and we'll just very simply talk about the testicles that are suspended from the body, and that's basically a temperature regulatory feature. The testicles also produce testosterone in the leg and the interstitial cells. This is the site of spermatogenesis, so we develop those sperm, and they are stored within the seminifera, moved and then stored. The epididymis stores a whole lot of sperm, and this is where it actually acquires its ability to fertilize as it becomes mature. If they're not utilized, they degenerate and are resorbed back. The vas deferens, simply the transport tubule from the epididymis to the urethra. There's really nothing else going on here. We'll store them until the time of ejaculation, and which, depending on the species, that might happen very, very quickly, and it might happen very, very slowly, depending on whether you're a bull or a horse or a ram versus a boar or a dog that does it quite slowly. Again, the urethra is right there, extending from the bladder through the pelvis and the end of the penis. That's a waste excretion, and the penis, male organ of copulation. And virtually every species has a little bit different variation in form. So again, here's a good cutaway. This is the reproductive organs of the bull. This reproductive organ of the pig, and I think you get the idea. A little bit different than what you see in the. Here's a good diagrammatic sketch. So if we look at bull sperm, you see the head of the sperm in the upper right, kind of a mid-piece as it attaches from the head of the sperm to the tail, and then all the way to the end. Very critical that you understand the different parts of the sperm is extremely crucial, because when we talk about artificially inseminating, and actually even in natural service, you can evaluate sperm for effectiveness and motility. If we do something to sperm and mess up the tail or sever the tail from the head, we can obviously have either a sterile animal or an animal that produces sperm, or we have a system in which does not result in healthy sperm being deposited into an animal, for you're going to have lack of pregnancies that way. So here's a couple of examples of abnormalities: a proximal droplet or a detached tail. Obviously, those animals, those sperm aren't going to effectively swim. A bent tail, obviously that sperm won't swim very effectively. I've also seen where you can shock them. I believe it's cold shock them to where that tail will bend all the way around its head, and that actual sperm will just sit there and spin in circles, and clearly you won't get it migrating very, very quickly towards the egg. Here's a cross-section of seminiferous tubules in bovine testes. It kind of demonstrates in the lower right one that you have a lot of sperm there that are advanced stage of being matured and are just about ready for actual use there. Male hormone function, very simple. When we talk about the male, we have just a couple hormones to really look at. We have luteinizing hormone, or LH, that makes those Leydig cells make testosterone, and FSH, which stimulates the tubules to nourish those spermatozoa we've been talking about. And as testosterone is made, it goes back to the anterior pituitary in the brain and tells the brain to make a little bit more LH and more FSH. So you'll see those three things kind of in cyclical fashion continue to work all together. There is a little bit of function influenced by photoperiod or day length here. So you have to think about which animals that you know of might be photoperiod breeders, or there's a breeding season, so to speak. And those of you who are deer hunters out there know that deer are photoperiod sensitive. There's a breeding season for deer, and it's really a response to uninterrupted nighttime in which they will be triggered biologically into breeding season and into heat. We get to female hormone function now. It's quite a little bit more detailed. Gonadotropin releasing hormone, or GnRH, originates in the hypothalamus. It then is transferred through the blood to the anterior pituitary, where again we're going to make LH and FSH. The blood picks up LH and FSH and moves it to the follicle, telling the ovary, hey, we need to make some follicles, make some primordial follicles, and keep producing eggs here. Which, as you remember from a couple of slides ago, that follicle is then going to make estrogen. The estrogen goes back to the brain, and we kind of keep that cycle going on until such time as which we have ovulation. Once we have ovulation, as you remember from a couple slides ago, that CL is going to form, and hopefully we have insemination occur, whether it's natural service or AI again, and we have a time hopefully where we have fertilization occurring. Well, the body has to give itself time for pregnancy to be successful. And for that to happen, the CL has to emit progesterone for a period of time, telling that animal, hey, I don't need to nourish another follicle right now. I'm pregnant. We have to feed a baby. So that CL will stay functional again as long as we have the presence, hopefully, of a developing fetus. The uterus is the pregnancy detection area. If the uterus never detects a pregnancy, the uterus then emits another hormone that comes into the game called prostaglandin. Prostaglandin goes back to the CL and makes the regression of that CL occur, to which we won't have any more prostaglandin, and then the reproductive cycle hormonally can begin again. Keep in mind, all this happens in about a 21-day cycle in most animals, and give or take a day or two, depending on which species that you're talking about. So we have ovulation, then levels of progesterone rise, and then let's say by about day 18 or so into the reproductive cycle, the uterus is going to be detecting or looking for pregnancy, and if it detects a pregnancy, then we just continue to make progesterone and we carry the fetus to term. If we do not detect pregnancy, then we need to get rid of the CL so we can ovulate another egg and have the opportunity to have pregnancy the next heat cycle. And we'll look at a diagram here. Again, all these activities make up that cycle. Again, the range is 14 to 29 days, and we can have the estrus behavior anywhere from just a few hours, and I listed 12 on the slide, but some of you that have dairy cattle know it might be just a couple, especially when it's hot, all the way out to 9 days. Like horses can exhibit estrus for about a week at a time. But hormone function can be altered, can be stimulated, whether it's via hormone therapy, whether it's via nutritional impact or whatever. I mentioned that you would see a diagram coming, and here is kind of a mirror diagram. On the top, you see what's going on physiologically. On the bottom, you see what's going on hormonally. So if we start at the left of the screen and come across, here's at the end of a heat cycle to where we did not have a pregnancy. You see a spike of estrogen, a spike of LH, and then at day 0 down at the bottom, that's ovulation. Hopefully that coincides within a few hours of insemination. Again, whether it's a natural service via a boar or a bull, or you go out with your AI gun and inseminate the animal. From that point in time, the developing CL, you'll see a rise in progesterone levels as we're right in the middle of the screen here. You'll see progesterone kind of reach its peak and hang in there for basically about from day 5 to about day 17. At day 17, the uterus is detecting for pregnancy. We have another follicular wave coming along that could be ready to ovulate if there's no pregnancy. So the uterus does pregnancy detection. If it detects no pregnancy, then we're going to emit prostaglandin, get rid of the CL, so we can have another heat at day 21 and start again. Now, if we detect pregnancy, then you'll see progesterone levels just stay high. We'll do a diagram of this in class if you're not quite following along with me. I understand this is a lot of conceptual stuff here at a pretty short amount of time. So we'll cover this stuff in more detail in class. So when we talk pregnancy, the onset of gestation follows the fertilization, and cellular division occurs about every 20 hours, and that period of attachment in the uterus is critical. Full attachment doesn't occur until about day 30 in cattle, and about day 21 in swine. And if we have no attachment that actually occurs, we call that early embryonic death. Now you start talking to your veterinarians, kind of the old standard of rectal palpation for pregnancy on cattle was about day 40 to 45 for the earliest times that they might want to palpate them by hand to check for pregnancy. You find some really good vets with really good hands can do it down about day 28 to 32, somewhere in that range. And obviously, the earlier you can detect pregnancy, the better, the more things that you can do strategically with your animals. So there's some advantage to that. The problems may come in to where you think you have a positive pregnancy in an animal, and when in fact then something happens and she loses the pregnancy, and therefore we miss the opportunity to perform something management-wise on them in order to cycle them back around. So that early embryonic stage is very, very crucial, and we don't often understand why we have early embryonic death, and there's varying percentages of that. I think in dairy cattle especially, because of stressors, the early embryonic death rate might be quite a bit higher than what we might expect. There's some species that it might be just a few percent. There's not a very good scientific connection that we can make statistically in what's going on there, but we do know that early embryonic death is a problem that we'd like to improve and focus on. Fetal stage then begins at about 46 days all the way through birth. About in the last section. Here's a picture of a developing bovine embryo at the 6-cell stage, magnified actually quite a bit there. You can kind of see what that thing looks like. If you've ever been with a veterinarian that's done embryo transfer, the eggs will look kind of something like this that they look at for health. Let's fast forward all the way to what we call parturition, which is the birthing process. The placenta responsible for maintaining that fetus and protecting it, exchanging nutrients and waste with the mother. And then birth is initiated with the release of a hormone called cortisol. So again, we're adding to our list of hormone things here. Hormone causes progesterone to decrease, estrogen to decrease, prostaglandin to spike, and especially oxytocin to spike, and that initiates some of the uterine contractions that begin the expulsion of the fetus. Relaxin comes into play, again, the kind of the last hormone that we'll talk about here. That is released from the CL, and that's what stimulates the pelvis to relax and kind of open up that birth canal. A difference physiologically in the male and the female. Us males, we have a fused pelvis down at the bottom, so there's not really a birth canal there. In females, there's a little elastic band there that's like a rubber band, allows to stretch so we can pass a birthing fetus right through that birth canal. So that relaxin will help flex that pelvis and relax that rubber band that is down at the bottom of the pelvis and allow that fetus to pass through the birth canal, hopefully safely. But all those things have to occur kind of in concert and do it the right way. And some of us are very good about letting animals go through the birthing process without interference, and some of us are very bad about interfering very quickly, and that's a discussion that we can have in class. Here's an example of an animal going through parturition, a mare that's going through the process here of labor. You can see the normal presentation of the head and front feet in the upper left picture, and then about halfway out here in the upper right picture in number B, and basically full expulsion of the foal there on picture C on the bottom left. And then right away, mama mare, she's doing her job, hops up and goes to licking that baby off and stimulating that animal to get up and nurse right away in the bottom right. That's ideal. That's how it should. Here's some normal presentation on the top diagram versus some, we have some problems in the bottom diagrams. And those of you, especially dairy guys who end up pulling a lot of calves, will understand that the bottom right one, the backwards calf, is certainly one that you can deal with. And then you have some other ones like the breech or the upside down or the head pulled back. Those are ones you better get your veterinarian on the phone and get some assistance, or you certainly run the risk of ending up with a dead calf or a dead cow on top of it. So keep in mind, be vigilant on normal versus abnormal presentations. And anything abnormal or calving difficulty or birthing difficulty, that is technically called dystocia, and hopefully you're tracking those rates at home. Here's a simple table that your book provides that gives you gestation length in days, and also the type of placenta that's there and the number of offspring that are usually born. So it gives you a good frame of reference. When we talk artificial insemination, again, you can't talk about the reproductive process without adding this in there as a management technique. And what it's done for the livestock industry is basically allow us to now select superior males to go with our superior females. And how does it fit in each industry, we can talk about, but clearly in the beef industry, it's a little bit of a tougher fit because they don't spend as much time out there, or their cattle out on large ranches or ranges, and it's much, much easier from a management perspective to run a bull with them. Dairy are a lot more confined, and so we'll more often, especially from a safety perspective, since we're working very closely with those animals on a day-to-day basis, spend more time artificially inseminating them. Artificial insemination success in horses is very limited and is not as common as some of the other industries. What are the drawbacks? Well, number one, when you go into AI, it's a lot. It is more expense. It takes more management time. It takes more labor. It takes more attention to detail versus versus natural service. The drawback of natural service is you have very little control genetically over what's going on. You select one or two or a handful of genetically superior bulls, that covers your herd, hopefully. And you aren't necessarily always sure about sperm quality unless you've had that tested. So it's not prominent in all of animal agriculture. Historically, it's recorded in dogs back in the late 1700s, and it really became prominent in cattle in the 1900s, and it was on the horses for a little while and then kind of lost its appeal. But basically, one bull will cover about 50 animals a year naturally. Some of you like to contend that number, but all the data supports 30 to 50 females is what a bull can effectively cover in a year's time. Whereas in a week, that same bull could have made 200 to 400 units for artificial insemination. And we can average about one calf for every 1.5 units of semen. So you can see a highly genetically superior bull can produce a lot of offspring in a hurry with AI. But the success of AI is really dependent upon the success of estrus detection. I can't stress enough. You'll hear me harp on it and harp on it. If you really want to do a good job with the reproductive program, improve your heat detection. That's the key. Breed more animals. Breed more animals at the right time. Everything else that's listed here, quality of semen, semen handling, high fertility, all those fall in line. But you really lose the game if you don't do a good job in estrus detection. Again, another chart on reproductive technology development. You guys can percent of AI matings. Notice just 8 to 10% in beef cattle, about 90% in dairy cattle, very common. Horses, it says 40 to 50%. That 40 to 50% would be on the high end, I would say, of the equine business. There's a lot more natural service kind of in your day-to-day areas. The swine at 80 to 85%, and then you see sheep and turkeys. Turkeys, 100% of the industry. A little on collection and processing. We're going to collect a donor bull with an artificial vagina, and if you've never seen that process before, they bring the bull out or boar and mate it on a dummy animal or a dummy apparatus and collect semen either naturally, manually through an electro-ejaculator. We'll collect boars a couple times a week. Boars can maybe every other day, bulls a couple times a week. And depending on the amount of sperm that they're making, more or less dictates that schedule. Here's a example, a longitudinal diagram of a artificial vagina. So you have a collection tube at the left end of the screen and a water bath and a rubber liner where you would insert that over the bull's penis in order to collect the donation. Obviously, this guy at Select Sires is really enjoying his job. You see the jumper bull there in the bottom and the bull that they're collecting on the top. Semen, once it's collected, is always highly evaluated by each of these semen companies, or in any they'll take it to their laboratory and check it for volume, concentration of sperm, motility of the sperm, and any abnormalities. If we have a problem, they're going to catch it hopefully right then and there. Then we do some extension of the sperm in order to kind of extend it out and mix it out into other with milk and eggs, and kind of make more units out of one collection. So we have about, in one normal unit of cattle semen, you're going to have about 10 million motile spermatozoa. Now, some of you following along in these industries know that we can make sex semen, and we can pull off the female, the XX, or the X sperm, and have only female sorted semen. And generally, that's sorted by weight, by the way. But in those straws, it's a quarter mil straw, and there's like 5 to 7 million sperm in those. It's about a half rate. If you're going to use it fresh, it only stores for about 24 to 48 hours if you're going to use it fresh. Otherwise, in liquid nitrogen at minus 320, and it can store basically indefinitely as long as there's liquid nitrogen in your tank. We can store it indefinitely without any loss in fertility. Other semen doesn't store as well. Bull semen you can store indefinitely. Boars, it's only modest success. Stallions, very limited success. So most all of those are used fresh. Again, I mentioned when we go to inseminating a female that estrus detection is really critical. All the other things fall in line. And you have to handle semen properly. You have to thaw it properly. If it's frozen, you have to deposit it in the appropriate area in the reproductive tract and avoid stress all the way through. Here's what we call a cane and some straws that are there. This would be what would be stored in a liquid nitrogen refrigerator or tank. And those half mil straws that are those 3 at the right side of the screen, those would each contain about 10 million bull sperm, and put in a plastic container there and into that metal rack and into the nitrogen tank. Here's a thaw unit and a breeding unit that you might see. You can see the guy's equipment box here on the right side of the tailgate. The blue thermos on the left is calibrated so that you can thaw semen at about 90 to 95 degrees Fahrenheit for about 40 seconds. That thaws it out, and then you see the AI guns laying in the middle of the tailgate. You can see them there. We insert the straw in there and be ready to go breed. Here's a diagram of someone artificially inseminating a cow. Notice we pass that AI rod through the vagina, through the cervix, and deposit right in the uterine body. A little on estrus detection. I've already mentioned that a couple times, but that signals the time of ovulation. What's the best indicator? The best indicator is mucus discharge. That's the best one. Right after that is an animal standing to be ridden. You can add a lot of other things on there. Other indicators might be she's riding, she's bellowing, she's restless, walks a lot. There's a whole lot of things. Technologies that might be used to help you: pedometers, cameras, KMAR patches or rub-off patches, and I think I've got a couple pictures here. Strategies that help you detect estrus. Just to set estrus detection time, morning and night, maybe it might be a good idea in order to go out and watch for animals in heat. Timing of insemination, it's very simply an AM/PM rule. If you see an animal in heat in the evening, breed her in the morning, and the opposite would be true if you see her in heat in the morning, breed her at night. So it varies a little bit with species, but that is kind of the general rule of thumb, that old AM/PM rule. Here's a heat detection patch that we can slap on the top of an animal. It's silver. When you place it on the tail head, it kind of sticks with a little bit of glue there. And then I think the next one, this cow's been ridden, and so it rubs it off, and so it's bright orange in color, and you can kind of look out across your pen and notice that animal clearly has been ridden. Now, obviously, you can have some false positives here. She could have rubbed on a gate. She could have been jumped accidentally. She could have hit it while she was laying down, and that get rubbed off. So it's not 100%, but it's a good man. Helpers out there as well. We'll kind of get towards the end of our section of reproduction here and talk about estrus synchronization. And what is that? Basically, we would get a group of animals together and synchronize them with hormone shots so that they would all come into heat about the same time. We would all breed them in the same time, so you'd have calves or foals or pigs all about the same time, baby pigs. We say, where is it the most appropriate and cost effective? Well, depends on your operation, what your challenges are. It's cost effective if you need to catch up on breeding. It's cost effective if you have a bit of labor shortage and can't do a lot of heat detection. It's cost effective if you're really bad at heat detection, because you can take that out of play. So there's a number of things to discuss on where it's appropriate and cost effective. I'm going to go through a couple of programs here just to give you an idea. There's a number of them out there. The first one is just the simple use of prostaglandins that causes regression of the CL. So if we go out and give a group of animals a shot of prostaglandin, if there's a CL present, they should be in heat within about 3 days after injection. So you can just simply watch them for that. Again, it's only present if there's a functional CL. So cows have to be between days 5 and 18 of their estrus cycle. And you say, well, she doesn't tell me, so how do I know? You're really not going to know unless you've really watched them very, very closely and carefully to know when the last estrus cycle was. That's a way to synchronize. There's a 1 injection versus 2 injection system of prostaglandin. And again, you have to time these things out if you're going to use a 2 injection system. Basically, if you use 2 injection, you inject them on day 1 and day 14, and so basically all the animals in your population should be bred or should exhibit estrus by about day 19, and you should be able to breed them, let's say, 76 to 80 hours after the second shot. Really works pretty well in well-managed herds. Okay, major risks with prostaglandin, don't forget, it will end a pregnancy. So if you have pregnant animals and non-pregnant animals in the same pen, and you go out and start shooting prostaglandin around, if you put it into a pregnant animal, you will terminate a pregnancy. So make sure that you are identifying animals properly, have good records, and know which animals are pregnant and which animals are not pregnant that should be getting shots. Next is what we call MGA and prostaglandin. MGA is melangestrol acetate. It's a feed animal that suppresses estrus in heifers. You feed it for about 14 days, pull it away, give a prostaglandin shot at about day 19, and you should see a group of animals come into heat then within 2 to 3 days. Conception rate can be above 80%. It's a really, really good program. I would say not widely used. The prostaglandin 1 or 2 shot prostaglandin system is pretty common. MGA and prostaglandin not so much. Then you get into this couple other synchronization programs, what we call Select Sync. Injection of GnRH. 7 days following, you go prostaglandin. 2 days after that, you go another shot of GnRH, and then you breed after the last injection, about 18 hours or so. 70% will exhibit. Co-sync is a variation of the previous one in which you give the second set of GnRH and breed at the same time. One of the things that you'll see in those two systems right there is animals may not exhibit estrus. It's important to understand that you have made them ovulate. The hormones should have been successful, all other things working properly, of course, that they should be ovulating. So it makes no sense, gang, as managers of an operation, AI technician, whatever, if you've invested the $10 to $15 or $20 to go through that protocol and then do not put semen in them, that's a waste of your time and a waste of your money. So if you're going to put animals in a synchronization program, unless something happened health-wise or it's too hot or whatever, whenever they get to the end of the protocol and you should be inseminating them, please inseminate them. That's good management. Can't stress that enough. Moving on, look at CEDAR synchronization, use of an intravaginal device that emits progesterone. Implant that thing for 7 days, inject prostaglandin on day 6, and then breed on estrus detection after you remove the CEDAR 3 to 6 days after injection. Again, very, very effective. I was actually on a farm that did a lot of the research on the time. PG600 used very, very effectively in swine, lowers the returning to estrus days after farrowing and schedules breeding there. And then there's actually what we call natural estrus detection. Actually works pretty decent in beef cattle and baby pigs. As you remove that calf or that litter of pigs, most of the females will somewhat suppress estrus while they're nursing young, at least for that early period of time. They'll focus on feeding the baby and not so much about getting pregnant again. If you take the babies away, then that signals to the body, hey, it's time to get pregnant again and make another baby. My baby's gone. That sometimes will signal a natural estrus synchronization to them. I would encourage anytime you get to thinking about doing any kind of synchronization protocol, get your calendar out, make yourself a map, make yourself a list of days, use your phone, whatever. Use some kind of strategy so that you know exactly what day I want to breed and work backwards from there in terms of when shots should be administered or when you should be feeding. Here's an MGA prostaglandin protocol. You're welcome to print this out and use it at home. Here's a picture of a CEDAR, an intravaginal CEDAR, controlled internal drug releasing device. Last couple things on this section, we talk about embryo transfer and in vitro fertilization. Embryo transfer is where we have an animal that's coming to heat, we've bred her, and we've put her through a process, a protocol that hopefully she'll ovulate multiple eggs. We breed her multiple times, and then we can go in there with a solution called flush, it's a saline solution, in which you'll essentially flush those eggs out and collect them into a collection tube, evaluate them for fertility, and then we can implant them into animals that are not as genetically superior, what we call donor or recipient animals. That way we can get more offspring out of our genetically superior animals. In vitro fertilization, or IVF, is where you actually go right in with a needle and aspirate or pick eggs, fertile eggs, right off of the ovaries. And then we put those in a petri dish and actually fertilize them in a laboratory. Can be very costly, obviously, because we're talking maybe $1,000 on the low end to $1,500, $2,000, $2,500, maybe even on the top end to do these protocols. Always performed by a veterinarian. I do know of a few lay people that know how to do this protocol, but I would recommend a veterinarian all the time that specializes in this, because again, you're going to invest quite a bit of money per protocol per cow. So make sure that you're ready to go and do this. Essentially, you've got to have genetically superior animals for your essential components, and then you need to have either a place to store those embryos in nitrogen freezer, or you need to have recipient animals ready to go in order to transfer those embryos to recipients. There's also things like sex semen and embryo splitting, and I say, what's the advantages or disadvantages of sex semen, which is a technology that's getting on 15 years old now, where again we sort X semen and Y semen basically by weight. I think the X's are a little bit heavier, just microscopically heavier. The reliability, so whenever we use sex semen, it's about 85 to 90% accurate. You'll hear producers say that's not true, but scientifically it's pretty well supported that 85 to 90% of sex semen users get heifer calves. And so that is used to help grow your herd internally without having to buy any animals. Bull calves in many instances, on dairies at least, aren't worth a whole lot, so there's no use in having them. Embryo splitting is a nice technology that's come along as well, and where you can take a embryo harvested via embryo transfer and split that thing in half, and then get 2 embryos out of one healthy one. And that's actually being used pretty commonly in both the beef and the dairy industry. Here's an example of that embryo sprillating process microscopically. It's fascinating to see that, and I do know a veterinarian that's pretty good at this and has gone through the protocol, knows how to split embryos, and she does it primarily on beef cattle. Some take-home messages in this section as we finish this up. Again, this is one of the most important and yet most difficult managerial challenges on virtually all operations. If you cannot successfully manage your reproductive process, you're probably not going to be in the animal business very long. So focus on this one right here and how to improve it from year to year. It should be a standing goal on your operation to have more reproductive success. Understand that process, hormonal function, and the timing of estrus cycle always will help you stay focused on how to maintain and improve your protocols. And know that the strategies you employ, know what you can do to improve reproductive success. Again, always keep this as a goal, and then always be looking for strategies to make it better. Whether it's more fertile animals, a better ration, better facilities, hormonal therapy, use of new technology, use of automation, sensors, cameras. There's a number of different things that you can use strategically in order to improve reproductive success. Again, one of the things I think you can really, as a producer yourself or as in a consultation role helping producers out there, really help them make more money is in this area right here. I look forward to our discussion in class, and thanks for listening.